called tracheids and vessel elements. And tracheids are pretty much ubiquitous across vascular plants. They're the ancestral type of xylary element in vascular plants in general. And vascular plants are formally known as tracheophytes. That's the technical name, and that refers to the presence of tracheids, which are the most diagnostic feature of a vascular plant. Remember, we talked about conducting tissue in some mosses, but mosses don't have tracheids, and tracheids are distinguished by having these secondary cell walls impregnated with lignin, again, like the sclerenchyma. They're also hollow at maturity, but unlike the sclerenchyma, these cells are actually involved in moving water through them, as are the vessel elements, which are a more specialized type of conducting element in the xylem that also has secondary walls impregnated with lignin, but they're broader in diameter typically. Here you can see a cross-section. This is a vessel element here. These are tracheids around it. And unlike the tracheids, vessel elements actually have openings, big openings in their end walls. So they actually have perforations in their end walls in what's called a perforation plate, or it can be completely open at the end, and these are connected end to end into what are called vessels. Whereas the tracheids themselves are connected by what are called pits. There are also pits on vessel elements, but all the tracheids have are pits, which are little regions of very thin primary cell wall that the water diffuses across. So there's no secondary wall in those regions, and water can move between the tracheids that way. Water can more freely move through vessels, both due to these openings as well as their greater width, but they're not as structurally, they don't impart as much structural strength to the wood as tracheids do, and they don't withstand quite the same tensions as tracheids. So in wood that lacks tracheids, which some wood does, it just has vessels, you'll have fibers in and around the vessels reinforcing them. And vessel elements are only found in flowering, well, I should say, they're best characterized from flowering plants and neophytes, that one group of gymnosperms that was once thought to be closely related to flowering plants. So these have evolved multiple times in at least a couple of different groups. They either supplement or replace the tracheids as conducting elements. All right, so are there any questions about xylem? We're going to go into how it functions a little bit more later. Phloem is another type of vascular tissue that has a totally different function. It also moves things through the plant. It's involved in moving substances through the plant, but in this case, it's the moving of photosynthate, of actual sugars, of organic nutrients through the plant. The xylem can move inorganic nutrients from the soil into the plant, but here we're talking about moving the organic nutrients that the plant has fixed, basically sugar solution. And the phloem moves that sugar solution from the places where it's produced, typically in the leaves, to the places where it's needed, which can be growing tips of regions undergoing growth in general, as well as places where sugars are being stored. And the cells that are most actively involved in the movement of these sugars are what are called sieve tube elements in flowering plants, or sieve cells more generally. And unlike the xylary elements, the tracheids and the vessel elements, these cells are living at maturity. So you might wonder, how could these living cells serve as conduits to move the sugar solution? Well, the answer to that is that they only take up a really small amount of the space inside of these sieve tubes. So they have protoplasts that are devoid of nuclei. They don't have any nuclei. They don't have organelles. They don't have ribosomes. There are no particles really inside these protoplasts of any size at all. And the whole protoplast just forms a little peripheral region around the perimeter of the inside of these cells, and the bulk of the center of this sieve tube is completely open. And it's open through these connections between the sieve tube elements, which are called sieve plates, that have sizable openings that you can see here that connect them. So the way that these cells can actually metabolically function without nuclei or organelles or any of these main types of machinery of a cell is because they're paired with another cell called a companion cell, which is their sister cell. So in the final cell division that gives rise to the sieve tube element, the other product of that cell division is the companion cell that has all those normal components of the cell, and it basically functions to oversee the physiology of that cell, that actually the sieve tube element. All right, so that's the general, these are the most important cells then in the phloem. And we'll talk a bit more about phloem function later, but one thing to realize is there's a really quick wound response here. If this gets broken open, you can imagine the leakage of sugar water out into the environment would be really detrimental to the plant. And the way that plants put a stop to that immediately is they have a wound response where they produce a substance that blocks up that wound instantly so quickly that it's incredibly hard to do anatomy on phloem, because as soon as you make the section, the wound response occurs and these sieve tubes are blocked off. So that's an interesting thing about their physiology that's really, you can imagine, important in terms of the plant's fitness. So the two types of vascular tissue are always found together. So the xylem and phloem are always found together in vascular strands, or within the vascular tissue in general. So they're discrete. Like here you can see the xylem. This is a leaf cross-section, lower epidermis and upper epidermis. And here you can see a couple of vessels in cross-section. Here's some tracheids. You can see the red stain here. That's the lignin in the secondary cell walls. Right next to it is the phloem. And here you can see a sieve tube element in cross-section, and there's a companion cell right next to it, always paired up like that. So the overall vascular tissue contains both xylem and phloem as discrete but adjacent elements, or adjacent types of vascular tissue. And the arrangement of the xylem relative to the phloem differs between leaves, stems, and roots, as we've already mentioned. All right, so now let's go to the kind of big picture of looking at how all of this is put together in terms of the overall plant body. So we're going to talk about the main organ systems first in plants. There are two major organ systems. There is the root system, which comprises just the roots alone, and we'll get to those in more detail later. And there's the shoot system, which takes in both the stems and the leaves, as well as all the modified, remember the flower is just a stem axis with modified leaves on it. So the important thing to realize, a couple of things, is the stems plus the leaves taken together have morphology, anatomy, and development that is distinct from that of the root system. And the distinction between the stem, the shoot, which makes up, of course, the stems and the leaves, as I mentioned, in the root system, the distinction between the shoot system and the root system is present even in the embryo. So we have an embryonic shoot tip and an embryonic root, the radical, in the embryos inside seeds. So that's something that happens early during development. So we're going to focus now on the different parts of the shoot system. So of course the stem, its primary function is to elevate the leaves and project the leaves up into the atmosphere where they can acquire light, and of course they're going to be in contact with CO2 as well. So that's the primary function of stems really, is really just to get the leaves out into the light. And the points where leaves originate on stems are called nodes. So wherever a stem or a leaf diverges from a stem, that point along the stem is called a node. And the region between successive leaves or successive nodes along the stem is called an internode, which just literally means between nodes. And then we look at the leaf. There are a couple of terms here as well. For most leaves have both a blade, which is the expanded portion of the leaf where photosynthesis occurs, should say the majority of the photosynthesis occurs here, and then the leaf stalk, which is called a petiole, which is just an unexpanded portion of the leaf that attaches the blade to the stem. The petiole, though, is part of the leaf. It's not stem tissue, even though it can look stem-like. It's anatomically, developmentally part of the leaf. And then on the upper angle formed by the stem and the leaf here, at the base of the leaf, we have what's called an axillary bud in most cases. And the axillary bud has the potential to develop into a new shoot, a branch, as you can see has happened here, where this has emerged. And this branch is morphologically and anatomically identical to the primary shoot from which it diverged. So plants have modular growth. They're producing identical modules through a process of branching, both in the stem, in the shoot system, as well as in the roots, as we'll see later, that can result in plants having a very different overall architecture from one another. Even a brother and a sister growing nearby one another have different numbers of branches, different orientations of branches, depending on the growth environment that they happen to find themselves in. Plants can't get up and move when the light moves to a different place. Unlike animals that can move around, plants have to be able to grow in different ways in order to adapt to the changing environment during their lifespan. And so this kind of ability to grow, have this kind of open development where you can produce modules in different ways that exploit the environment or utilize the environment in different ways is something that plants can do that animals can't. And I should mention at the tip of each one of these shoots is what's called the apical bud. And it's at those apices that we have the production of new stem and leaf tissue. But this point is now about open development as opposed to the kind of closed development that we have, this determinate growth. I mean, our bodies are determined to have a certain form at maturity, such that you and I don't differ that much. We all have 2 arms and 2 legs. Normally, we can move around. We don't have to grow in order to adapt to our environment. Plants have all kinds of variation in terms of the way that they grow. And an important distinction here in the way that they grow is that their growth is really indeterminate or indefinite. Those points from which growth occurs, those embryonic regions that growth occurs from in plants, they never become fully differentiated. Plants retain embryonic tissue in their bodies throughout their lives that allow them to keep growing indefinitely. And that's very different from animals. So the areas that have the potential, that contain embryonic cells that retain the potential for continued growth are called meristems in plants. And in primary plant growth, these meristems are found, the types of growth that result in elongation, that is, elongation of the shoot and elongation of the roots. These regions are found at the tips, at the apices of the shoots and the roots, and these are called apical meristems. Okay, and these apical meristems are where we have active cell division during primary growth of the plant body. And again, this can continue indefinitely through the life of the plant. So if we look at a shoot apical meristem, we'll get to the root later. The area of active cell division is up here in this dome-like region at the very tip of the shoot. And you can see it's darkly staining. These are embryonic cells. The shoot apical meristem is primarily undergoing cell divisions that are parallel to the surface here. It's producing cells in a downward direction mostly that's resulting in basically movement of this apex upward as it produces cells downward. You can think of a bricklayer standing on top of a brick wall and putting bricks down as he or she is building that brick wall. And that's kind of comparable to what's going on up here. So the apex is moving away from cells that it produces. And these cells then will elongate and enlarge to some extent as they're produced before we get down to a zone where primary growth is finished and there's no further division or elongation. At the periphery of this shoot apical meristem, we have the production of what are called leaf primoria. So all leaves are produced out at the periphery of this shoot apical meristem. And here we see a couple of new leaves. We can see here where a couple of leaves were projecting out of the screen or to the back of the screen that were 90 degrees in their orientation from these ones. And here we have a couple of leaves that are even older out here. Okay, so that's the general way in which we have development here. And you can see these darkly staining regions in the axils, the upper angle of this developing leaf and the stem what are called bud primordia that will become these axillary buds from which new branches can form, new shoots can form. And these will remain embryonic throughout the life of the shoot, or at least potentially, and can at some point be activated to form a branch, a new module. All right, so now I'm going to show you a little film clip that basically goes beyond that one that I showed earlier of the seed germination occurring. This is after germination is completed, and you're going to see the apex of the plant moving upward. And keep an eye on the leaves that are down near the base, and you'll notice that as this process occurs, the inner nodes can elongate for a little while, but then that stops. And those lower leaves are not going to change their position. They remain just where they are. So here we go, and you can see the apex is moving upward, but that leaf is being left behind. There's no more growth going there. So this shoot apex is leaving behind mature stem below it, and once that's fully expanded and primary growth has ceased, the height of a given node is not going to change throughout the life of that plant. Some people think that plants grow from the base upwards, from the surface of the ground upwards, but that's not true in the vast majority of plants. Grasses are an exception, and since we're used to cutting lawns, we have a tendency to think of growth that way. We'll talk about grasses as an exception later. Growth in general of plants is from the tip downward. All right, so when we look at these 3 types of tissue in the shoot, or any part of the plant, the dermal, the ground, and the vascular tissue, these tissue systems develop from distinct cell lineages in the apical meristems. So these really aren't classifications of convenience. They reflect cells that have very different origins, but they all originate from the apical meristems. Here we can see ground tissue in this region is formed from ground meristem. In this region, below the dome, the dermal tissue, the epidermis, is formed from cells that are only along the periphery, at 1 cell layer in thickness around the shoot apical meristem, and they remain strictly peripheral as mature cells. And then the vascular tissue forms from these embryonic cells, or initials called procambium, which just means before cambium. All right, so that's the general development in the shoot apical meristem. Are there any questions about that before we go on? Okay, so you've already seen the cross-sections like this in cartoon form and in lab. I just want to review, though, that the stem and cross-section looks very different between a eudicot and a monocot. Again, we have cortex and pith that are clearly differentiated in a eudicot stem because we have the vascular bundles arrayed in a ring here. Whereas in monocots, the vascular bundles are scattered throughout the stem, and we don't really have the ground tissue differentiated into pith and cortex, so those terms really aren't used in the description of the anatomy of a monocot stem. But they are important for understanding kind of the topography of cells within a eudicot stem. When we look at the overall plant morphology, the arrangement of the leaves on a stem can vary between species. This is something that's usually taxonomically fixed. I mean, it can be conserved even within some plant families, which are a pretty high rank in classification. And the way that the leaves are arranged generally tends to minimize the shading of the leaves by one another. One type of arrangement is called alternate, which means that there's one leaf diverging per node or per position along the stem. This is a really common pattern. If you look down from the tip of the stem downward, or the shoot downward, this often looks like a helical or spiral pattern, such that there's minimal shading of the leaves by one another. In other species, we get two leaves diverging per node, directly opposite one another like this. And that's called opposite phyllotaxy. Phyllotaxy just means leaf arrangement. Phyllo is leaf and taxis is arrangement. And at successive nodes, these usually diverge at 90 degrees from one another. One node will be like this, the one above it like this, and the next one like so that they're not shading one another. And then finally, whorled, which is a term we introduced in talking about the plant parts of the flower, is 3 or more parts per node. And this is relatively uncommon in the vegetative parts, but is what we see throughout flowers in general, usually 3 or more parts. Okay, we'll go on to plant morphology of the shoot next time before jumping into the roots. Okay, so see you next time.